afternoon, everybody. Thank you very much for being here. My name is David Gil-Johan. I'm presenting on behalf of the Exicure team. Exicure is OTC traded in the U.S. under the ticker symbol XCUR, and as such, please note I'll be making forward-looking statements, and I'm not here to sell stock. So on slide 3, a couple quick facts and figures about Exicure. We're based outside of Chicago on technology that was developed at Northwestern University in the laboratory of Chad Merkin. We are, again, trading since about May of this year with 44 million shares outstanding, about $100 million so far invested into the company from a notable set of tech luminaries. Refer to our website if you want some more information about that. We currently have 3 drugs in clinical trials, 2 in dermatology, 1 in oncology, and I'll share highlights of each of those programs with you here today. Our last raise was done at the end of August. It was a $22 million pipe, which brings us cash into September into 2020 of next year. The team that we put together is one of a very high science group. In addition to myself and the CFO, David Snyder, the former Chief Scientific Officer of IDERA at Kambarkhanamala, the former Chief Executive Officer of Moligen, Matthias Schroff, and Jocelyn Trokenheim, who leads up BD from Takeda, are all part of our team. The technology, again, came out of Northwestern University in the academic labs of Chad Merkin, who is a luminary in the field of nanotech, Shab Thaxton, our MD advisor, David Walt, the founder of Illumina and currently a professor at Harvard, Helen Kim, who was most recently at Kite prior to their acquisition by Gilead, and Jay Venkatesan, who's an investor in the Boston-based area and spent the majority of his career at Brookside. So we have 3 general ideas here at the Exicure table. We are advancing our technology in inflammatory diseases, genetic disorders, and cancer. Our proprietary platform I'll talk to you about today is called spherical nucleic acid. So our oncology lead asset is called AST-008. Our oncology asset, we just reported out data in September from our phase 1 safety study in healthy volunteers. That is a TLR9 agonist, which we expect to launch our phase 1b/2 here before year end. On the inflammatory disease front, we have a local topical approach to using nucleic acids, and I'll share with you highlights from our Exicure 17 program, which reads out this quarter. And then in genetic disorders, I think this is one of the exciting areas we're looking to expand into in 2019. You'll see here some side-by-side comparison we generated last year in neurology, showing side-by-side comparisons with nusinersen from Spinraza. We have also shown that the platform works in GI, lung, and an eye. So if you have partnership interests in those particular areas, those are places where Jocelyn's actively looking to expand the platform partnership potential. So what is a spherical nucleic acid? Our spherical nucleic acid is a 3-dimensional arrangement of DNA or RNA. So it's the short DNA or RNA that you know and love. Think siRNA, microRNA, antisense molecules, etc. But what we've done is arrange them around the outside of a nanoparticle core to form what we look like is a little bit of koosh ball. So the DNA is very much on the outside of the nanoparticle structure, and that's very different from what historically others have done in the nucleic acid space. What we do at the end is have a nanoparticle that has about a 20 nanometer core size, and the oligo shell around the outside contains about 80 nucleic acids. And again, those can be DNA or RNA-based molecule. So why should you care? On this slide, very simply, the architecture makes a difference when it comes to delivery into cells. And so on the left-hand side, what you're looking at is the classic challenge in the field and the paradigm for the last 20 years, which has been how do you get DNA or RNA into the cells? And what you can see on the left-hand side is keratinocytes, and they don't take the cells don't take well up the DNA. On the right-hand side, what you can see is a 3-dimensional architecture of that same DNA. So equal molar concentrations, equal number of DNA molecules, just arranged in 3-dimensional format. And there you can see the cells light up. And all we've done is gone from a 2D architecture to a 3D architecture. And again, the take-home message from a lot of fundamental science that has been done is that architecture makes a difference, and the cells like the 3-dimensional structures, and the 2-dimensional structures not so much. We know quite a bit about how these get into cells and how they ultimately can function in different biological systems. There's about 100 or so academic publications on this technology now. Quite simply, the 2 major questions that I'm asked is how do they get in so uniquely? The answer there is scavenger receptors. So the scavenger receptors turn out to uniquely address these 3-dimensional architectures and allow an endocytosis process to go onto the cells. And the second question that I often get is how do you possibly protect them from nuclease degradation along the way? In other words, how do we get them into the cells intact when we've so nakedly exposed them on the outside of the nanoparticle? And the answer there is that we've actually created an architecture which is inhibitory to enzymatic degradation purely based upon the sterics and the electrostatics. So packing them densely on the surface not only leads to internalization from the scavenger receptors, but also keeps those pack-bound-like nucleases from being able to chew them up. So we find that on a nucleic acid per nucleic acid basis, we not only get now the cellular entry, but stability of any nucleic acid sequence goes from 10 to 100-fold higher, simply based upon the architecture-based effect. So why should one care about better delivery into cells and into tissues? If you look at the nucleic acid space, you're going to find 90% of the drugs have been developed after liver-based targets. At Exicure, what we find is that better delivery leads to broader applications. So here we're looking at localized applications of nucleic acids. So think gels and creams on the surface of the skin, topically applied drops or injectables into the eye. We've shown that oral formats work in GI type settings, and in the immuno-oncology space, we're looking at localized injections into the lymphatic system in order to activate an immune response. So anywhere your inside touches your outside, or you have an ability to inject locally, we now think we have an opportunity to use nucleic acids where they haven't been used before. So far beyond a cool science idea, by this point we've built a clinical stage pipeline. The ones in dark blue on slide 10 have for you the clinical stage assets. Immuno-Oncology, again, our AST08 project is currently in a phase 1 trial, launching a phase 1b2 trial here before the end of the year. Derm, our Exicure 17 program, is an anti-inflammatory IL-17 receptor molecule being tested first in patients with psoriasis. That one will have some early indications here this quarter. And then finally, we have a partnered program with Purdue that's an antisense molecule against TNF-alpha. So some highlights from each of these. The Immuno-Oncology platform, for those in the room, you'll be familiar with the idea about cancer immunotherapy and that being a Nobel Prize winning idea as of late. But the idea on slide 13, of course, is how do we make those therapies better? And the answer is to take cold tubers and make them hot. And so we want to activate an immune response, and we want to use that activated immune response in combination with the checkpoint inhibitors. How does that one do that using DNA or RNA? The answer is toll-like receptor pathways. In particular, our AST08 product is a TLR9 agonist in three-dimensional form. TLR9s have had a great year. If you've seen presentations from ASCO and AACR, Dynavax, Idera, Checkmate, and others have announced some great combination data with TLR9s plus their checkpoint inhibitors. So why should we care about the Exicure platform? Basically, the idea is that getting into cells, again, makes a big difference when it comes to potency. So by getting into cells better, we activate toll-like receptor pathways better. We can lower the dose. We have a better tox profile, and we can expand the routes of administration. So we can go beyond intratumoral injection. We've actually been looking at subcutaneous as well as IV routes of administration for our TLR9. So on slide 15, just one snapshot of over 124 different preclinical animal experiments that have been done that demonstrate this effect. And I've chosen this slide to show you the difference between the linear structures, again, and the three-dimensional architectures. And in this case, the red line, which we're showing you, is a historical breast cancer growth curve that we tested from TLR9, Coley's Pfizer's CpG 7909 sequence. When we take that same sequence and arrange it in 3D format to make our spherical nucleic acid platform, you can see with the Exicure technology, we go from having almost no response to having a complete response. And that's simply, again, going from a two-dimensional structure to a three-dimensional structure. Otherwise, it's the same nucleic acids, same sequence. So again, hammering here the point that architecture makes a difference when it comes to potency. This is via a subcutaneous delivery in a breast cancer model. We've shown preclinically that this works in breast, lung, melanoma, colorectal, and bladder cancer. It works sub-Q, IV, and IT. It works in combination with CTLAs, PD-1s, PD-L1s, etc. We took this into a phase 1 trial here in the UK, the idea being that we wanted to collect safety and tolerability data, but also look at activation of NK and T cells, and then compare that against historical benchmarks in the TLR9 field. Happy to report last month we successfully wrapped up that trial, and we saw about a 3.5-fold activation of NK cells over baseline in a dose-dependent manner across the volunteers, and we saw about a 10-fold activation in T cells, setting the stage for us to take this into a phase 1b2 trial in the US later on this year. We expect to enroll about 30 patients in that phase 1b2 trial and begin that trial in combination with Pembro. So we'll be looking for an intratumoral injection of our AST08 in combination with both Pembro naive and Pembro refractory patients. Types of tumors that we're looking at, so basket tumor in this case, we'll be looking at head and neck, Merkel cell, cutaneous cell, squamous carcinoma, and also expanding to any palpable tumors. So in the few minutes remaining, wanted to explain why this platform is also important in genetic disorders and show you a side-by-side comparison with a molecule that you might be familiar with called Spinraza or nusinersen from Biogen. And here's an animal model that was run by the Ohio State University, and the red line here on the survival plot is showing you a single-dose injection of nusinersen in a spinal muscular atrophy model in mice. And what the investigators found is when they took that same nucleic acid and just arranged it in 3D, forming the Exicure version of that nusinersen, what you can see in the blue line is the extension of survival. There's about 4x increase in survival, and all we've done is take the nusinersen, the commercially available Spinraza drug, we've designed that in 3D, and again, you can see that the architecture significantly impacts the potency. Recently, we also announced at the Lyric conference the biodistribution data that went along with this, which shows a reason why that we get such better potency. And what you're looking at on slide 19 is a biodistribution study in the rat. The oligo here has been labeled fluorescently so you can see it. At time equals 0 on the left, the 2 rats there you can see have been injected with an equal molar concentration, again, of DNA, either in the 2D format, nusinersen, or the 3D version of it. By 24 hours, the middle 2 rats, what you can see is that the nusinersen has started to leak out of the spinal cord and the CNS and has ended up being cleared by the body, and you'll see it there in the liver, kidney, spleen. The Exicure technology, by contrast, stays in the CNS, and you can see it throughout the spinal cord and the brain, even out through the 168 7-day period. So we're really excited about this. We're obviously seeing favorable PK/PD profile, again, by using the Exicure version of a known nucleic acid molecule. This opens up for us tremendous numbers of opportunities in genetic disorders. We're focusing this quarter on looking at neurology, DMD, Huntington's, ataxias, Batten's diseases, et cetera. Any place where nucleic acids or genetic targeting, we think that this technology could play a huge role in advancing treatments and cures. Inflammatory diseases, the last bit of our protocol I just wanted to show you here is our program called Exicure 17. Exicure 17 is a local topical application of an antisense molecule directed against IL-17 receptor being currently applied in patients with psoriasis. And the idea here is the penetration is so good at the cellular level that we can simply apply the DNA or RNA-based Exicure technology in the skin. In psoriasis patients, it's the penetration deep into the epidermal and then ultimately the dermal layers. The plaque test that's going on currently in Germany just wrapped up. 21 of 25 targeted patients were enrolled and completed in the trial. It allows us to use 5 different sites on the same patient, which gives us a positive and negative comparator, and also a low, medium, and a high dose of our Exicure 17. At the end of this study, the patients are giving us punch biopsies from each of the sites. From the punch biopsies, we'll be able to look at the messenger RNA level. And so quite simply, from a phase 1 trial, we're able to look for the genetic markers that showed that we were able to hit the targets, which we were able to design in the laboratory based upon our SNA technology. When you look across the space, some comparators to look at, we think that Exicure is interesting and unique in the space in that it is not married to one particular biology. Again, we work with siRNA, antisense, splice correction oligos, TLR9s, et cetera. And so rather than say we're limited to one of those areas, the Exicure platform, again, is an architecture-based story which allows us to access unique tissues and apparently improve the potency in all of these different areas across the nucleic acid space. Our IP portfolio continues to grow, over 60 now issued or allowed patents and 120 that are pending based upon this 3D spherical conformation of the nucleic acids. So in summary, Exicure has a proprietary platform based upon our 3D spherical nucleic acid technology with 3 drugs currently in clinical trial. Our oncology, you can watch for a phase 1b/2 launch and announcement before the end of the year. Our inflammatory disease protocol will read out here this quarter with the topical psoriasis patients, and watch for an expansion in the genetic disorders, again, using that data that we generated side-by-side comparisons with nusinersen. On the final slide, slide 26, happy to take any questions or be in contact. My email is davidg@exicuretx.com, or you can reach our CFO, David Snyder, at dsnyder@exicuretx.com. Thank you very much for your attention and for being here today.